Right now I'm getting my packed cell volume and my total protein, so I have my EDTA tube right here. I'm just going to mix this 4 to 6 times, make sure that all the anticoagulant is thoroughly mixed with the blood. And then I have my hematocrit tubes. I've got 2 of them right here that I'm going to fill. So I'm just going to take the cap off of my blood tube, and then I will take my hematocrit tube, or my capillary tube, and I will just let the capillary fill the tube, pull it up. I'm filling it about half to 2/3 of the way full. And then I'm going to clean off the end and put a clay plug in it so that when I centrifuge it, it doesn't come out or spill. Okay, and then I'll fill my second capillary tube. Fill it about halfway. And then I'll clean off the outside of the tube and put the clay plug in. Okay, once I have my 2 hematocrits and they're filled and plugged, then I'm going to come over to my hematocrit centrifuge, and I will take the lid off of this, and I'm going to place my 2 capillary tubes in across from each other to balance each other out in the centrifuge with the clay plugs pointing to the outside. And then I'll put the lid back on. And this is going to make a loud noise when I start it, so I'll go ahead and turn the camera off for that. But I'm going to let that spin for about 3 minutes, and then we'll come back and check our packed cell volume and total protein. Our centrifuge has finished spinning now, so I'm going to pull out my hematocrit tubes and get my packed cell volume and total protein. So I have my 2 hematocrit tubes in here, so as I pull them out, I'm just going to take a look at them and make sure that the plasma is normal colored. It's not lipemic or hemolyzed or icteric at all. So it's a normal, very pale yellow, kind of clear color. It's not pink or yellow, which would be what I'd be looking for with those colors. So what I'm going to do now is I have my hematocrit capillary tube reader right here. I'm going to line up the tube with the red blood cell column onto the 0% line, and then I'm going to slide it down until the top of the plasma lines up on the 100%. And then where the buffy coat falls, that line in between the red blood cell column and the plasma, is where my packed cell volume is going to be. So this one falls at about 41%. So that's what I'm reading on my card, about 41% with this one. And I'm going to go ahead and verify that, and I like to just look at both tubes to make sure they're consistent with each other. So again, I'm lining up the red blood cell column on the bottom and the plasma on the top. And this one reads also 41%, so they are the same. Okay, so after I have my packed cell volume, I'm going to record that over here, 41%. I'm then going to take a microscope slide, and I'm going to score my hematocrit tube right on the buffy coat, and that's that line in between the red blood cells and the plasma. I'm going to make a little score on that so that I can snap it and then put the plasma on the refractometer. So I'm just going to do a little cut. Then I'm going to gently snap it, be careful of the sharp edges. And then I'm just going to let the plasma drop onto my refractometer. I'm being careful to not scratch the prism on this refractometer, but still get enough on here to be able to see. Okay, when I have a big enough drop, I can then close the cover. And then I'm going to hold my refractometer up to the light, and I'm going to hold the cover down, and I can see there's going to be a blue line on here where the lines intersect is where my total protein is going to read. I can twist the eyepiece to zoom it and focus a little bit better. And the total protein reads at 6.3. So I'm going to ask my supervisor, just come take a look at this, make sure. Do you have a second to check my total protein?
Absolutely. And that reads at 6.3.
Thanks. And then once I'm finished, get my total protein. I'm going to record that over here also on their record. Then I can just rinse off my refractometer, prism and the cover, and then dry it off. And then I have my packed cell volume and my total protein value.